Hi, Farhana.
Hey.
Hey. Okay, so I'll start and then I'll pass it on to you in a few slides, okay?
Okay.
So we'll give it just a few more minutes. We have about 5 more minutes. And then once we start, we're going to try and cover the introduction to bioinformatics and genomics. So hopefully we'll do it faster than in an hour or 2 hours.
Yeah. So in the morning today, when I sent the email, I sent to only those people who filled out the forms. So that it doesn't go like to 1,000 people, you know?
Yeah, that's good.
So it's around like 55 of them.
All right, so we're going to be starting here in just a couple more minutes. In the meantime, I just want to remind everyone that we have a page where you submitted your registration and you should have gotten that Zoom link. But for people that just now submitted, I see a couple of people submitted just now. I hope that they will be getting the link in time as well. So, okay, so maybe to begin, I'll just introduce a little bit the project and then I'll pass it on to Farhana to say a couple of words about what we're going to be doing today. So Omics Logic is a bioinformatics education portal that was designed to address some of the needs in bioinformatics education and data science education across the Louisiana Biomedical Research Network and the National Association of Idea Principal Investigators. One of our objectives is to develop resources that are easy to use, specifically designed for biologists and non-bioinformaticians. And we hope to learn today about the interest in a variety of different applications of bioinformatics including several different types of omics data. So the program was recently announced at the Southeastern NEPI or Southeastern IDEA meeting. And here you can see on the slide, Dr. Gus Kasulis, who was announcing this project and making sure that we have faculty as well as students. So this is designed for both undergraduate and graduate students, and that everyone knows that this is available for free. It's going to be covered by different institutions that we'll discuss today how the registration is going to work. But the key point is that this is going to be available for free for those that are either from LSU, Louisiana Biomedical Research Network, or from any NEPI project. So to give this a little bit more introduction, I will pass it on to Farhana. Farhana is a postdoctoral student at LSU. And so she'll briefly talk about these programs and registration, right? And then I'll kind of cover what's going to be in each one of these programs.
Hi, everyone. This is Farhana. I am a postdoctoral researcher in Dr. Kasulis' lab in Louisiana State University. So this semester, we are offering these 3 courses. One is the Introduction to Bioinformatics. That is for the very beginner. It's good for undergraduate students and whoever never had any bioinformatics course before. The second course that we are offering is the Genomics and Metagenomic Data Analysis. It's an intermediate level course. And afterwards, for those who are very interested and have previous knowledge about bioinformatics, they can take the advanced level course, which is the Data Science for Biomedical Data and Intermediates. So in the flyer that we sent you earlier, and can I share my screen? I don't know whether I can share my screen, Ilya.
I think you can now.
Okay, so can you see my screen now?
Now you have to press on share. Did you do that?
Okay, so this is the flyer. Okay, so most of you already received this flyer. And in the flyer, we have a link here that actually shows, if you click on the link, it will take you to the website, and you will see that there is a form that you have to submit so that we know that the people are interested in this course. And once you submit this form, we will send you notification and other information about the courses. So this is what the course schedules are. So the first course, the Introduction to Bioinformatics, starts in January 24th, that is next week. And it's pretty much a month-long course. The second course, the Genomics and Genomic Data Analysis, starts on the 25th of January, and it's a little bit longer than the first course. It ends on the March 22nd. And you can either take the Genomics or the Metagenomics Data Analysis. And the last course starts on January 27 until April 21st. So before you take these courses, you have to register through LSU, more specifically via the Gene Lab. So to register it, first of all, you have to have an iLab account. So after this orientation session, I will send you all the links and important informations on how to register. So first you need to create an account in the iLab or the Gene Lab. The people who are already in the LSU vet school, you guys already use the iLab for utilizing some of the instruments. It's a core facility for us. Whoever doesn't have the account, they have to sign up. And sometimes some of the students already face this issue that their PI's names are not on the link on this system. So for those people, they have to like inform us their PI name, email address, and phone number so that we can include the PI in this system. So once you sign up for an account, you will see a page like this. And then I will scroll down here. I'll scroll down here and you have to create an account and then you sign in. And after you sign in, you will see on the left-hand side, the core facilities. You have to click on that core facilities, and then you will see the CDER core facilities and services. You have to click on that. And after that, you will see a page like this. And then on the right hand side, on top corner, you will see the request services. So it will take you to the training module. Once you click on the request service, it will take you to the training module, and you can see and select like what are the courses that you are interested in, whether you are taking the one course, the first course, or all of them. You will select those courses. And then right now you don't see it here, but the updated system will have the names of the courses. Then you can select it. Afterwards, you have to put the phone number. If you are outside the LSU or the LBRN program, then you have to have your specific phone number for us. Otherwise, you just write LSU or LBRN, and then we will put the account number here. So after that, you will just have to submit the request to us, and we will go through all the information, and then we'll accept it, and then the registration will be complete. So after this orientation session, I will send you all about the informations and everything, and the procedure how to register, so that if there's any confusion, you can look at it. And after that, if there's any other questions, I will be happy to help you. Just contact me. That's it, I guess, Ilya.
All right, thank you, Farhana. So again, this was just a little clarification about how you can register. And what I will do is go over the actual content of this program to really explain what are the differences between these the three different programs. So as Farhana already mentioned, we're going to have these three programs: Introduction to Bioinformatics, Genomics, and Data Science. In fact, here you can see that it's going to be either genomics or metagenomics. We'll talk more in depth about metagenomics in our next orientation session, but today we're going to just cover Introduction to Bioinformatics and Genomics. So before we begin, I already posted a link in the chat. This portal is where you're going to be able to access all of the material. So the registration that Farhana mentioned, this is to get yourself registered according to the three different options. Either you are from LSU, and then you have to register through iLab and mention that you're from LSU, or if you are from LBRN, which is Louisiana Biomedical Research Network, then you also register through iLab. For everybody else, you can just directly register on this Omnic Logic portal, and within the portal, you'll be able to specify which institution you're from. So just make sure that you are mentioning you're using your email address from your institution, and then if you're a part of NetBee, mention that in your bio. So once you go to this link, again, this link is in the chat, you will see an option to create an account. And so this account is going to be important to use, and I'll explain in a little bit why and how that is going to be used for certification. Once you are registered, you will be given several options, and those options will include some basic access or some advanced access, so don't choose those options. We will be sending out a follow-up email, and if you have registered and you want to participate in one of the programs, we will be reaching out to you. If you're a part of NetBee, That's going to be, again, free of cost. So at this moment, you can take a couple of minutes and just create an account or log into your account, and I will show you where in this account you will be able to use different areas to get access to this upcoming training. So on your account, once you're in, and again, all you need is your email address, or you can use your social media or something like this. Please update your name. And if you are a part of NEPI, if you are in a lab, you're a student in a lab, or you're a PI, just mention it right here under your description. There's going to be a little section for bio. That way we will know to reach out to you with the right information. After you have completed your profile, we will be using this profile to track your progress and recommend additional training that you will find on the portal. And I will go over this training today. So you will, after completing certain requirements of the program, you will be able to receive the certificate. And that certificate will be issued based on where you came from. So either you will get a certificate from LSU, and it will be signed from someone from LSU, or from LBRN, Louisiana Biomedical Research Network, or from NEPI, the National Association of Idea Principal Investigators. Okay, so I'll just give it 2 more minutes. If you still have not created an account here, please do so now. And if you have already, maybe you can mention that in chat. Again, I'm just getting notifications about people registering, so I'm just going to give it a couple more minutes. Okay, so once you have registered and you have, again, added your name and a little description here about yourself that should include whether you are from a specific institution, part of NEPI or LBRN or LSU, please mention that right here. All right, thank you. And if you have already, if you mention this in the chat box, that would be very helpful as well. So first of all, how do you get started? The first program that we're going to discuss is called Introduction to Bioinformatics. So if you have minimal background with bioinformatics, you don't really have any experience, maybe you have read and you have heard and you're interested in it, but you don't really have any practical experience, this is the best place to start. Now, you will be able to register for multiple programs. For example, you can take Introduction to Bioinformatics first, and then once you complete it, you can go on to the more advanced programs. Those advanced programs could include all 3 of the other programs: genomics, metagenomics, or data science. But first, for those that don't have any background, we recommend getting started with this introductory program. Now, this introductory program includes several modules, and these are Introduction to Bioinformatics, Bytes and Molecules, Bioinformatics in R, and Bioinformatics in Python. So this will really orient you with some terminology and some basic skills for you to get started. It is typically something that you can complete in about a month's time. So that's why we're offering for those that complete Introduction to Bioinformatics to continue on to the more advanced programs. As soon as you have registered, you will already have access to all of these materials. Some of them are completely free, and some of them will have some free modules that you can just test and see if that's something that you would like to do. So these are found, and again, I'll just quickly switch here to the portal. Once you're logged in, sorry. So once you're logged in, for example, this is my profile. You can see here information about myself. And then if I click right here on courses, I can scroll or search, or right here, you will see getting started. So if you click on getting started, you will see these modules that I was just showing on the screen. Introduction to Bioinformatics, Bytes and Molecules, Bioinformatics in R, and Bioinformatics in Python. So these are some of the starting materials that we will cover. Now, beyond that, what we will do in the program is we will use these basic modules to see the applications of bioinformatics, to learn about the terminology that is commonly used with bioinformatics methods. We're going to do a little introduction to coding with Python and R, just so that you're aware of what goes into some of these methods of analysis. And then we will provide you with access to some introductory projects. So these projects are going to show you how people that have used bioinformatics in a variety of domains, like infectious diseases, oncology, and others, have been able to use the methods that we cover in these programs to achieve research results. So what actually went in there? So the way you can find those is, first of all, there's the search field. You can search, for example, for Ebola, or you can look over here on tags, and these tags include some typical types of materials that you will find. And down here, we have organized them into these sections for getting started, sections in R, sections in Python, projects, etc. So the one we just discussed was getting started, which is right here. Now, beyond that, once you are getting comfortable with how things are organized and you understand how to navigate the site, you'll be able to find a lot of additional materials that you can just try and do on your own asynchronously. So let's talk briefly about this omics logic programs in general first, and then we'll jump into introduction to bioinformatics. So as a part of this initiative, we have collected materials from a variety of researchers that address these specific types of applications of bioinformatics. So first of all, introduction, a general overview of bioinformatics, genomics, transcriptomics, metagenomics, epigenomics, and data science. These will be offered throughout the year, including in the summer, a specific program around a research fellowship. And this research fellowship will provide you with an opportunity to work on an independent project. So the idea here is to provide training with basic skills, and then to offer a variety of applications, including oncology, infectious diseases, neuroscience, agriculture, biotechnology, and others. So what typically goes into a training program like this are a collection of resources that we will be providing for those that participate. So first of all, you will have access to the training materials. Those training materials will be completely asynchronous. You will be able to find them on this portal, but you will also have a program page that will organize these materials into a sequence. Once you complete each one of those, you will also be sent a video that you can watch that explains how the different elements of this program are combined together. You will also have access to a big data analysis platform that you can use to analyze public domain data. And some of those public domain data sets are going to be explained in the materials that you will be provided. You will also find elements of code that you can get started with to apply what you have learned in R and Python, and some explanations on how to do that. So what does the syntax mean? What does it do? Some examples that you can try right in the browser, and an explanation of how you can take that and use it on your own system. Now, the previous programs that we have run, we try to address the needs of those that want to learn about, but also apply what they have learned to their current research and to projects, sometimes citizen science projects, sometimes research projects. And we have quite a number of people that were successful at completing the program and learning about how to apply these methods in their research that they were a part of. So what we want to cover in this first program, the Introduction to Bioinformatics, is to provide a general overview to those that don't have a lot of experience already with bioinformatics. So this will include bioinformatics and big data, how big data and the need for computational technologies is changing bioinformatics, including an introduction to a variety of omics types of data, and then to methods of analysis, the basic statistical analysis, as well as machine learning for a variety of these types of omics data. We will touch on the scripting aspect, but that will really be minimal. It's not intended to be in depth about any one particular language or application. It's really to provide you with a sense of what bioinformatics is and how to apply it in a general theoretical sense. And so hopefully this is something that anyone with any background can participate in and benefit from, just learning about the terminology. So the three main things that we will cover are informatics, statistics, and biology. And so what that means is that we will talk a little bit about what kinds of information technologies, what kinds of statistical and analytical methods, and what kinds of applications of bioinformatics are important to understand, and how that is changing different fields where bioinformatics is used. So primarily, we will try to talk about this from the perspective of biology and data. So how does data describe biology? And how can we study biological phenomena using different types of data that provide us with this detailed view of what's going on? But we will also talk about the applications of bioinformatics in clinical and different industry projects as well, specifically in biomedical application to diagnostics and selection of therapeutics or discovery of therapeutics and things like that. Now, we will cover several different types of omics, including genomics, epigenomics, and transcriptomics, but very superficially. So there won't be any in-depth explanation in this particular Introduction to Bioinformatics program. It is just going to provide you with the sense of what does this data describe and what does this data look like. To understand what it looks like, we're going to also cover several publicly available data repositories where you can explore projects that have been submitted by researchers across many different types of labs, and you will be able to find a variety of different project examples that are typically associated with a publication. So hopefully you will find some relevance to whatever field you're involved in. Now, as I mentioned, one of the challenges in bioinformatics is being able to not only learn about bioinformatics, but practically experience what bioinformatics can do for you. And so to introduce you to different methods of analysis of bioinformatics, we have a platform called the T-Bioinfo platform that is color-coded with explained methods that allows for a variety of inputs and outputs, and really will provide you with a guided process to understand the logic of bioinformatics. So for those that are getting started, this is the best way to go from general understanding to practical application quickly, right? And hopefully that also reinforces the logic of why am I doing what I'm doing and what am I expecting to get out of this process that we call a pipeline or a workflow. So to summarize, the Introduction to Bioinformatics program is going to explain how big data is used in bioinformatics or biomedical research. We will talk briefly about, the applications of bioinformatics. There we go. Different types of technologies, and some concepts of data science that are going to be useful as you continue to explore what bioinformatics can offer. And so to see a detailed curriculum, you can find the detailed curriculum on this landing page. And so I'll just show you this landing page, and I'll paste the link here as well. So you can find this information right here on this link. So if you scroll down, you will find here the full syllabus. So if you have any questions about the syllabus and what it includes, please don't hesitate to reach out to us. But this is the full overview of the program. Again, it's fairly short, but it's going to be good for those that are just getting started with bioinformatics. So this program, we ran it in the previous semester as well. We have quite a number of students, including graduate and undergraduate students that have completed this program. And it really can take a biologist, somebody that knows nothing about bioinformatics, and help them understand why they should learn this or how to apply it. And at least will be useful to read some papers that include bioinformatics, or if you're going to submit a grant and you're going to incorporate bioinformatics, we'll provide you with better terminology to interact with your bioinformatics core and kind of let them understand what exactly you're trying to do. So the other programs, again, these were beginner programs. The other programs that we're going to talk about are project-oriented. So project-oriented means that we're expecting those that have completed the other type of training, not the Introduction to Bioinformatics program, but genomics, metagenomics, and data science, to be able to submit, to learn skills that will allow them to produce a project of their own. And that's really the best validation that you're able to apply what you have learned. And we hope that you can do it in a fairly short time. So to do that, part of this Omics Logic portal is an opportunity to, once you complete your profile, to be able to submit a project right on the Learn portal and apply what you have learned in this training to a data set, describing everything that there was to that you did, and really focusing on reproducible research. So as you complete your profile and you learn by acquiring points by completing different coursework, then you will be able to submit this project that we that I will explain in a second. But that's the major differentiation between what we call a basic program and an advanced program. So the advanced programs are really oriented for those that want to use this for a particular project. And so I'll return to this towards the end, but let me briefly describe what will be covered in the genomics program, and then I will go back to describing what the project-oriented component really is. So for the genomics program, which is an intermediate program, we will cover the basics of genomics, the applications of genomic sequencing data analysis to study a variety of different examples like germline mutations, genetic diseases, cancer, and similar things to that. So briefly to describe what I will do today is I will cover some of those basic things, provide you with some examples, and then at the end, I will go back to this idea of project-oriented training that you can learn in these programs that are going to be coming up. So as you all know, DNA, especially the human DNA, is very complex, and it's organized in such a way that we can understand some of it, but really we need to have all the information about the genome to be able to ask questions about the DNA. Not only that, also the DNA itself is stored in a very compact way. And so to understand what information it contains, we need to understand the organization of the DNA, which is organized by chromosomes. And so the chromosomes are essentially these strings of DNA that are attached to histones. And when we think of DNA, right, the first thing that we need to think about is what are the actual elements that matter, right? Where should we look at? And so where we need to look at is typically referred to as the whole exome, right? So what are those specific regions of DNA that produce protein genes? So what we'll do in this program is we will talk about whole exome sequencing that helps us understand how the genotype, or what's written in the DNA, defines the phenotype of an individual cell, the phenotype of a tissue, of an organ, or the whole organism. Now, it turns out that only 1% of the human genome, about 1 to 2% of the human genome, are protein coding genes. And that number has significantly went down from the original expectations that researchers had when they were starting to sequence the human genome. So today we have about 22,500 genes, but that number varies across different research fields. And so really, we're going to look specifically at that known part of the genome, which we call the exome, right? The part of the genome that is being expressed. One of the basic applications of genomics is to identify bugs in the code. How can we link specific variants or differences from the reference that allow us to make assumptions that the sequence change results in a particular change in phenotype? For example, here you can see how a mutation could change the cell phenotype, which in this case is the red blood cell, to be the sickle cell, right? So that sickle cell disease is defined by one such change. What we will also look is how we can start characterizing these mutations and start understanding their functionality, how individual nucleotides form nucleotide codons, and how changes in specific positions in these tricodons result in a change at the amino acid level. We will talk about different types of point mutations, like substitutions, insertions, and deletions. And we will discuss what are genetic variants versus mutations versus neutral mutations, pathogenic mutations, and essentially mutations that don't matter. And the way that we will talk about this is we will try to understand the changes at the sequence level, at the DNA nucleotide level. We will then try to understand what are some of the thresholds that are used to understand whether the mutation is a coding or a non-coding mutation, where that mutation is found. Is it found inside the gene, at the promoter of the gene, at the splice junction of the gene, or whether it is silent? And we will also look at the impact of the non-coding mutations and whether these non-coding mutations matter at all. We will also take a look at some examples of where such germline mutations are known to play a difference in phenotype, in how they affect the phenotype. So when we talk about these types of mutations, we will also apply this to somatic mutations as well as germline mutations. And we'll talk about how to find them and what type of sequencing is required to be able to find them in with a level of confidence that you can actually annotate them and interpret them. So genomic variation that we will discuss will include looking at individual single point mutations, as well as structural rearrangements and copy number variation as well. And as I already mentioned, we will take a look at how we can observe the impact of these mutations in the sense of affecting a phenotype. And that could be either a disease or that could be just a difference in condition. We will also take a look at the relationship between the specific known genes that typically have these mutations at different levels. At the level of where they are found in the chromosome, what does the gene look like in terms of what's being expressed and what's the variation of expression, at the 3D structure of the protein that that gene codes for, and also at how people validate that these mutations actually cause the phenotype or a change in function. Now, to understand all of this at the practical bioinformatics level, we will also discuss how this data is generated. So we'll take a look at different types of sequencers, and different methods of sequencing, including targeted sequencing, amplicon sequencing, and whole DNA sequencing that is applied for genomics. And to really understand the process, we'll take a look at how the sequencing machine works and what type of data does it produce. And when we come to that part of the data, we will actually look at what do these files look like. And so we'll try to understand what is the information that is encoded in this DNA sequencing data, and then how can we analyze this data, which we will start from by learning the logic of analysis. So really, how can we perform alignment? How can we find variants? How can we interpret the variants structurally, and how we can measure relationships between sequences and changes in the sequences. So here are some of the applications that we will look at. We will look at the need to clean or pre-process our data. We'll talk about the need for calling a variant, determining whether this is a mutation, and annotation. How do we actually know what this mutation could be related to? But we will also look at additional applications of different methods where we can either visualize the data that we have or identify a particular type of mutations like germline or somatic mutations. We'll learn to work with genome-wide association studies where we need to understand the relationship in a population to a phenotype and what is needed to make that kind of an association. And we will also take a look at downstream interpretation of the found mutations. So how can we actually know what a particular individual mutation, or multiple mutations, what do they mean and how can we identify other factors associated with that? And so here as an example, we will use a study that shows how we have individual or monogenic SNPs and how we can look at polygenic SNPs, where monogenic are strong associations that are typically rare, but they are commonly associated with the phenotype, or polygenic scores that are more complex. And how do we actually analyze the data to try and determine what is associated with what? And this is typically referred to germline mutations. So some of the things that you will learn in this part of this course is you will look at how to load the data, how to process the data, how to find meaningful associations, and also how to provide a report on what you have found using both the Bioinfo platform and coding in R. Now, beyond those, we will also look at some of the applications of genomic sequencing for biomedical research. And there we will look at examples for drug metabolism, somatic mutations, inherited mutations, rare genetic disease, as well as drug resistance in bacteria, which is going to be a little bit different, but with similar principles. As a part of that, you will learn what goes on behind those typical reports that are provided to cancer patients, for example, and how do these studies that intend to understand the mechanistic or statistically significant mutations, how do they use this research to actually produce such results? Also, we will look at how cancer occurs, and we will use some examples, the commonly known and something that everybody has probably heard about, like P53, as well as less known as and maybe less understood individual mutations or multiple mutations that have high association with a specific phenotype. So we'll talk about point mutations, insertions, deletions, and rearrangements like copy number variations. So I also mentioned that other applications of sequencing might include finding specific mutations responsible for drug resistance, like antibiotic resistance and tuberculosis, or association between variants identified in short sequences. For example, we'll take some examples of viral sequencing data to try and find specific mutations that have been associated with the phenotype. As a part of this training, you will get access to our online coding, which is like an element that you can find inside the coursework, where you can practice how to write code with a lot of guidance, right? So one of the important parts of this program is to provide you with the logic of analysis and then introduce you to practical skills that you can also take with you and continue to learn on your own. So when we will do that, this is just an example of an exercise where we will take several DNA sequences from patients, compare them to the wild type, and try to determine what should be the specific diagnosis based on this information that we should give to a patient. So again, some practical examples of how you can use this type of data and this type of analysis to identify some important information for biomedical research. So in summary, we will provide you in this genomic program, Omics Logic Genomics program, a variety of examples and opportunities to try to analyze whole exome sequencing data from humans and non-humans alike. We will introduce you to how this type of analysis could be applied in basic and translational research. You will understand the terminology and the commonly used approaches for analysis, and will be able to perform those independently as well, using the platform for logical and user-friendly analysis, as well as learning how to take the process data and extract from there meaningful insights using some coding skills, not a lot, but at least some, in R, and we will also show you how to deal with a variety of different data types that you will be expected to work with as you are introduced to genomic data analysis. So to briefly summarize, the two programs that we discussed today are Introduction to Bioinformatics, getting started with big data bioinformatics, and Genomics. Now, I think tomorrow we're planning to do another orientation session that is going to be dedicated to metagenomics and data science, which are more advanced programs for those that already have some basics in those fields, like you might have some background in microbiology, or maybe you're interested in data science, which will typically require you to have some background with coding. So you must have at least a little bit of experience coding in R or Python, because very quickly programs will become challenging for those that do not have that background. So again, how do you register for these programs if you're interested? This is the page for Introduction to Bioinformatics. So again, I put in the link in the chat for Introduction to Bioinformatics. And this is a page for the Genomics program. So the Genomics program, the Genomics program right here. These are the topics and the syllabus. So you will find here all of the specific details. All of these programs are going to last for 3 months, so for the whole semester. The Introduction to Bioinformatics is much shorter in terms of what we typically expect people to spend on that program, but you can take up to 3 months to complete it. Genomics and metagenomics and data science might require a little bit more time. The process will be that you will have all of the materials. Like for example, this is a course on this portal that you registered for already on the Omics Learn portal. So here you will find some asynchronous materials, and every week you will have an email with a video lecture that connects these materials into a program. In addition to that, you will have access to our mentors. So if you have any questions along the way, you're struggling with some piece of code, you're trying to build a pipeline and it's not working, or you have a conceptual question, you would like to understand some topic that you don't think was explained well enough, we will have 2 Q&A sessions per month that you can join to ask all of those questions. Now, Farhana is going to be the course coordinator. She's going to be available for collecting those questions and making sure that we have those questions ready for whenever we have the Q&A session. And she will be able to point you in the right direction for registering for those Q&A sessions. What does a program actually look like? Let me just I'll go here. So a program will have, for example, let's go to this genomics program. So this program, as you can see, will have, once you're registered, you will see here the full curriculum, and you will be able to see here the video recordings that are sent to you as well, and complete those. And once you are signed up, you will find on your profile that right here, you will find whatever program that you're a part of. You will find that mentioned right there. For example, here, this is a program that already finished. And once you have finished, you will find your certificate of completion right here. And also you will be sent a PDF version of that program at the end as well. So now for those of you that are from LBRN, LSU, or NEPI, maybe Farhana, you can take a little bit more time to just go over the process again. How do you actually register yourself if you're from LSU or LBRN?
Okay. Let me share my screen one more time. Okay. So most of you already saw the flyer and already filled out the forms to us. And we have the names and informations about you. And now, like afterwards, you have to register for the courses. And it has to go through the Louisiana State University Gene Lab or the iLab. So this is the way that, so first thing is you have to have an account in the iLab. So most of the people like who are in LSU or the LSU School of Veterinary Science who have used the core facility, you all have the account in the core facility. However, the people who are outside Louisiana State University or maybe in the LBRN or somewhere else, you have to sign up for an account in the iLab. So I will send you the link to sign up. After this orientation session, I will send out an email with the link and will help you out how to create an account here first. So some of the students, undergrad or graduate students already reached out to me and saying that they don't see their PI's name in the system. So for those of you, if you don't see your PI's name in the list, just send me an email and with the informations of your PI's name and email address and also the phone number, we will add your PI's name in the system. Then you will be able to register. So first, after creating account, you will have to sign in and then you will see a page like this. You will have your username and password. You will sign in and afterwards you will see a page like this and on the left-hand side, there will be a tab called Core Facilities. You have to click on the Core Facilities. And then you have to select the CEDAR Core Facilities and Services. And then on the right-hand corner, on top corner, you will see the request services. So this is one of the services that the core facility does. So you will select the training modules under the request services tab. So once you get in there, you have to select the courses that you want to take. So we already updated the name of the courses for this semester. So you will see all these three names, the Introduction to Bioinformatics, Genomics, Metagenomics, and the Big Data Analysis on the left-hand side, and you will select, and if you have any specific comments, you can write in here. And then you can save this form and submit it later, or you can directly send it and submit it with the after introducing the fund number. So those of you who are part of the LBRN program or part of LSU or School of Veterinary Science, you can just write that here, and this course is free for you. So we are gonna put some numbers here and it will be free for you. However, the people who are outside Louisiana State University, they have to put their fund number where we should charge for the courses. And then you just submit the request to us. So then we will go through the overall information here, and then we will accept it. And that is how your registration will be completed. So I will send out this information in an email afterwards, after this orientation session. And still, if you have any questions, just send me an email. I'm physically here in the School of Veterinary Science on the 3rd floor. Whoever in LSU or in the vet school, if you guys want to see me or have any specific questions, you can see me or you can send me an email. I will try to help you in the registration process or any other questions if you have about the courses. So that's all, Ilya.
Okay, thank you, Farhana. Again, for those of you that are at LSU, you can find Farhana or contact her. The same for LBRN. And then if you're a part of NEPI and you're from another institution, you can directly sign up and you can just mention in your profile which university you're from, and please use your domain name from the university, not Gmail or something like this. So in addition to this, I wanted to mention that this orientation meeting has been recorded. You can find it right here. So you will be able to review any of the program description that we kind of shared with you today. We already shared with you the links to the individual pages if you're interested in learning more about the topics that are going to be covered. So I'm just going to put that again in chat. This is the Omics Logic Genomics, and this is the link to Introduction to Bioinformatics. So if you have any additional questions about the topics that we're going to be covering, we'll stay here for another few minutes to answer those questions. And for those of you that are interested in metagenomics or data science, we invite you to join us tomorrow at the same time. So we will review those as well. Yeah, if you do have any questions, you can put them in chat. And if not, again, thank you for joining today. You will, maybe Farhana, you can add your email in the chat. So if anyone has any questions, they can reach out to you. So yes, the courses are going to be asynchronous because people have different schedules. Farhana, I think you just sent that message just to me and not everybody. But if you do have questions, that are going to be twice a month an interactive Q&A session. That Q&A session will help us address any questions that you might have. So any technical questions or theoretical questions, we'll actually have trained bioinformaticians here that will be able to answer your questions. Okay, do we have any programs on how to handle isoform transcriptomic data? Yes. So we have, again, an asynchronous course called Transcriptomics. In that course, we discuss all from the beginning to the end, the transcriptomic data analysis approaches, including single cell transcriptomic data, how to work with isoform transcriptomic data, and others. And if you have any specific questions that you want and you're already familiar with the general terms of transcriptomics, feel free to reach out to me. My email is elia@pine.bio. Okay, so Charles is asking, I work in a hospital lab facility and not yet taken any post-grad program. Please, what does the program have for me since I'm on the bench for now? Though I seem to be fascinated by bioinformatics and data science. So we recommend starting with the Introduction to Bioinformatics. Again, Introduction to Bioinformatics, maybe I'll just share my screen once again, just briefly. I'll show you where you can get started and kind of explore what direction is most interesting to you. So if you go to the Courses tab, you can click on Getting Started right here. So if you click on Getting Started, you will see these are some of the basics that you can begin with. And if you have any additional interest, you will see some examples in the Introduction to Bioinformatics, and specifically here in the Bytes and Molecules course. So in the Bytes and Molecules course, for example, you will find here applications of bioinformatics to precision medicine, to space omics, to other things. And you have some introductory materials on genomics, transcriptomics, metagenomics, protein structure and function, etc. So this is a course that you can explore if you're just getting started. All right, well, again, thank you everyone for joining today. We will be sharing this recording, and if you do have any additional questions, please feel free to email us. And tomorrow we'll cover the metagenomics and the data science program. So if you're interested in those, please join us tomorrow at the same time. Thank you, and we hope to. Yes, it is the same Zoom ID and password for the meeting tomorrow, I believe. If you have been getting the emails, Harris, then please check those emails from Sonerika. Excellent. Okay, thank you, Charles. Okay, excellent. Thank you, Farhana, for your help, and for everyone else that joined today. and we'll see you again tomorrow.